So, do you see my screen?
Yes.
Yeah. Okay. Thank you. So, as Jasmine just told you, I'm Virginie Calderon. I'm the manager of the Bioinformatics core facility at IRCM. Our principal goal is to help researchers and students with the NGS data analysis. So we are performing, all the types of analysis regarding RNA-seq, Ataxic, ChIP-seq, whole genome, whole exome. But since 3 years, a lot of people are interested in single-cell experiments. So we have developed skills for such analysis. So today I'm going to present you an introduction about the single-cell experiments, the diversity of protocols, so what you can do with this type of data, how to start from the biological point of view, and also the basic bioinformatics analysis, and also how to get to the advanced analysis like data integration, trajectory inference, and pseudotime analysis. So the principle of single cell is to be able to dissociate a cell from maybe a tissue, an organ, and capture the transcriptome of the cells. So then you can compare the transcriptome, for example, of all the cells, and maybe detect some differences between the cells from maybe the same organ. So there is a lot of things you can do with a single cell experiment. So for example, you can characterize and identify heterogeneous cell populations. So that means you can maybe identify subpopulation of cells inside a tissue. You can also discover new markers or a new regulation pathway. So for some biological question, it could be interesting to see if something is regulated differently in some conditions. You can also discover new cell types, new cell states or rare populations. So if you are doing bulk RNA-seq, you will not see this type of signal, but by doing single cell experiment, you will be able to detect small populations.
Sorry, Virginie. We just see your presentation on the first slide. I don't know if you.
Oh, I'm moving. Okay.
Okay, perfect.
Okay, okay. We'll let this like this. Okay. Also you can go to more advanced analysis to build hierarchies between the cells. If you have different time points, for example, you can extract some knowledge about how the cells differentiate during the time. So this is especially interesting during the development. So you can identify relationships between the cell lines. And of course, at the end, you can also have, an atlas, a profile for each tissue and organs, depending on the condition, from an healthy donor or in a specific context like disease. So here I put some example extracted from the 10x Genomics website. So today I will talk a lot about this technology because this is the one we are using at IRCM. But there is a lot of protocols to do single cell experiments. So here in this example, they used the single cell experiment to link a disease to a cell subtypes. So in this case, they have identified 7 known lung cell types. Here they are listed, but using a single cell experiment, they were able to detect a new subset of cell. Here you see this is really, really small. That represent 0.5% of the cells, so it's a really, really rare cell types, and they were able to link this to a phenotype in the case of the cystic fibrosis patients. So that's something you can do with single cell, is to detect really rare cell types, contrary to bulk RNA-seq, where you just have the mean signal of the transcriptome. So this is the next example in the case of a breast cancer. In this case, they wanted to describe the subtypes of T cells in this cancer, and they were able to develop a signature that has a significant impact on clinical outcome. So that's something you can also extract from single cell experiment. It's signature that you can use later to do prediction on patients. And in this last example, in the case of the lung cancer, they wanted to have an overview of all the subtypes identified in the lung cancer. And in this case, it was used to identify new targets for immunotherapy. So again, another way to use single cell data and experiment to develop new things. So a lot of people are mostly doing single cell RNA-seq experiments. So the main advantage of this technology compared to the bulk is that you can identify more finer transcriptome changes compared to a bulk signal. Of course, there is advantages to the single cell. Of course, it's the identification of transcriptomes at the cell level, but the cons is the experimental cost. For now, it's really expensive, so you are not doing a lot of samples at the same time. So you maybe start with 2 or 3 samples, and you see what you get. And also because with the single cell experiments, you need the more advanced bioinformatics skills. In the opposite, for RNA-seq today, you can have a tool like Galaxy for the one that don't know how to code. They can use this environment, this friendly environment, to analyze RNA-seq. But today, for single cell experiments, you still need a bioinformatician to produce at least the first step. So before talking about bioinformatics, I will talk a little bit about the good practices for this type of experiment, because as a bioinformatician, you will have to know this information. You need to know what is the sample type. For example, are you working on nucleus or cells? This is not the same thing, because if you're working with the nucleus, if you are doing a single cell RNA-seq experiment, you will have mature RNA and immature RNA. So if you are doing a pseudotime analysis or velocity analysis after that, you will not have the same signal that using all the cell. Also, you need to know what is the cell line. Are you working with organ, with tissue? Because we know that there is a lot of issue working with some tissue if the dissociation was not done well. So it's always important as a bioinformatician to know where the data come from. Also, there is protocols now to work with frozen or fixed cells. The quality is improving, but still it's better for now to work with fresh cells, fresh living cells. But for some research lab, it's important, they have the data, the cells are already frozen, so it might be interesting for them to try this technology on these important samples. Also for the preparation, of course, if the biologists work from tissue, it's really important to be careful during the analysis about the doublets in the experiments, so you can detect if during the preparation of the library, if more than one cell was encapsulated. So you will have to use some tools maybe to detect such bias. Also, as a bioinformatician, if they have performed a cell sorting before the sequencing, you can check if the sorting was done well. So if they tell you which which protein was targeted at the surface, you can just check at the end that this is what you get at the end in your experiment. So again, you need to talk a lot with the biologist to know what was the experiment. And also, we advise biologists to remove dead cells from the experiment, because this will add another bias in the experiment. If the RNA is free in the samples, it will add ambient RNA. So we would have to remove this, and sometimes you cannot remove it because we have too many ambient RNA. So the quality of this type of experiment is really based on the quality of the sample. So that's why there is a big emphasis on this. This is really, really important. If the cells are not in the good states, you will not have good data. So that's why you need to be concerned about this. So also, before starting anything, you have to be really prepared for this type of experiment. So usually people are working with fresh cells. If you are in Montreal, you need to find a place to do this type of experiments. So if you want to use the 10x chromium, you need to find one that is available in Montreal. So there is several chromium in Montreal. I think it's more than 10 right now, but not all of them are available to as external services. So, for example, at IRCM, you are free you can come and ask for the services, but there is also other institutions like maybe Genome Quebec that is offering this service. So why it is important? Because between the end of the cell preparation and the beginning of the chromium experiment, there is a maximum of 30 minutes. So again, it's a lot of preparation. You cannot send your samples by the post. You need to be there to have a chromium near you to do that. So this is the first step: find a chromium or find whatever system to do this. Then the sequencing, of course, and you need to think about who is analyzing the data. So the first thing people are doing is to find a collaborator. So a collaborator is free, but a collaborator has not a lot of time, and a single cell analysis is taking a lot of time. So that's not always the best way to go. So you can take a bioinformatician to do it as a services, or hire someone, or it can be you. So maybe that's why you are here today. So you can try to learn this process. So to do this by yourself. So if you want to do it by yourself, you have to think about computing resource. So the first step of the analysis require computing power, so you can use a local server, or of course compute Canada resources. And at the end, you need to think about the cost of all of this. It's around $3,000 per sample for all the molecular biology. And if you want to use bioinformatics services, the price started around $1,000 per project. So you need to think about all of this before starting anything, because it's really, a step-by-step preparation, a lot of synchronization, and also found someone that is available. I'm talking about the bioinformatician, because if he's not available, you will not have what you want. You need to discuss with it. You need to be sure. Either the biologist or the bioinformatician, they need to be close to each other and discuss a lot about the project and what the data means, because bioinformatician solo cannot say everything, and the biologist solo cannot do what the bioinformatician is doing. So this is really the NGS data that need the more close collaboration. So about the experimental design, so that's something always important when you talk about NGS data. So in this case, for the number of samples, it really depends on the budget. So if it's your first single cell experiment, I advise to start with 1 or 2 samples at a time. I have seen a lot of people doing 1 or 2 samples, and at the end the cells are dead, or the count was not right, or the sorting was done really bad. So you do not start this type of experiments with 5 samples, okay? So you start small, and then when the protocol is working, you can do more and more samples. So this is 1 or 2 samples. This is not a lot, but also you need to think about bias. So for example, in human, you have a lot of variation between, for example, male and female. So if you are starting to do this type of experiment, you can think about including both samples in the experiment. And also, contrary to other NGS experiments, it's not mandatory to do biological replicates. So it's not like in RNA-seq where you need 3 replicates, for example, or sometimes in ChIP-seq, ENCODE will recommend 2 replicates. In this case, if you are doing 1 or 2 samples and find something, it's already something important, meaningful. Also about the number of cells, that's something the bioinformatician need to know. What number of cells are you expecting? Because the 10x chromium experiment is targeting a number of cells. So you are telling the molecular biologist, I want 5,000 cells. So at the end, we should expect this number of cells. If you don't have this number of cells, that means there is something wrong. Maybe your cells were dying, or something clogged in the chromium, etc. So you need to know what was the target at the beginning. So with the chromium, you can maximum target 10,000 cells at a time. So usually people are doing 5,000, but sometimes they go at the maximum. So depending on what you want to capture, if you are interested in a rare population, it might be interesting to go at the limit, at the maximum. But if you just want to know the proportion of some subsets of population, you might need just 5,000 cells to be able to capture this information. Also, if you're working with an homogeneous or heterogeneous population, it would be different. If you just in a homogeneous population, you need less cells. In heterogeneous, like I said, if you want a rare population, you need to go to the maximum. Also, to target a population of interest, you can use the cell sorting, for example, with the FACS, but it's really it can be really tricky because you can have a mortality level higher using this FACS. So you need to train before doing this type of experiment to be sure that at the end of your process, your cells are in a good shape. Then for the recommendation, you need to check at the beginning of the analysis that you are at the recommendation for the minimum number of reads per cells. So 10x is recommending 20,000 reads per cells. And if you want, to have deep sequencing, we are talking about 50 reads per cell to capture maybe lowly expressed genes. So the target, the bracket is between 20 and 50,000 reads for this type of experiment. So that's something the bioinformatician need to check first. Do you have enough information? Do you have enough read to pursue the analysis? So everybody is doing single cell transcriptome, but there is other protocols and other options available now. There is also the single nuclei Ataxic experiments. So it's to capture the chromatin accessibility at the cell level. So it's another way to look at the regulation of genes that may be complemented by single cell RNA-seq. So you can also combine these 2 experiments on the same cells. So you will have the information for one cell, the information of the transcriptome, and the information of the accessibility of the chromatin. So that's something new at 10x. So now this is really interesting to link the expression with the accessibility. Also, for all the immunologists, they propose a protocol for immune cell profiling for the VDG antibodies, so they can capture which VDG are present and the quantification of this immune cells. Another option is to add cell surface protein capture. So if the biologist can choose an antibody that is specific to a population, you can add a layer over the expression to be able to say, okay, this cell population has this type of protein at the surface, so it's this type of lymphocyte, for example. So because the transcriptome of the cells, you can see maybe some markers specific to lymphocytes, but the protein surface is more easy to capture if the gene is not a lot expressed at this time. So that's another layer you can add, is protein surface to identify the cell populations. So yes, for single-cell transcriptomics, this technology was developed now more than 10 years ago. It has evolved a lot in the last years, and it was at the beginning with a drop-seq. The drop-seq has a lot of advantages, like it's a most customizable method, so you can have an in-house drop-seq. The SmartSeq has the advantage to give you the possibility to capture the full transcript, also the full RNA. But now the 10x Chromium is the most standardized method. So it's really easy to compare experiments done with the Chromium. There is less technical bias in this. So that's why now people are mostly using this technology as a service in facilities. So it's an emerging technology. I see that in 3 years we have moved to from RNA-seq to single-cell experiments. So a lot of people want to do single-cell experiments, and sometimes we need a lot of education because it's new and people don't know what to do with all of this data. So that's why today I will present, what we can do with single-cell experiment. So the principle of a single-cell experiment, in the case of the RNA-seq, is to use beads in the Chromium experiment, is to use beads that have at the surface, sorry, a barcode that is unique. So inside this, all the beads have a different barcode. There is, I don't know, I don't remember, but it's billions of barcodes possible. So this will capture the mRNA with the poly-A tail. So that means that you exclude from the analysis all the other RNA that don't have poly-A tail. That's something that you need to know, because sometimes the researcher is interested in something, and this RNA, maybe this non-coding RNA, is not captured in the experiment. Okay, so be careful. You have to know that the RNA are captured by the poly-A. So it's a poly-A capture, and this barcode will allow you, after the analysis, to say, okay, this transcript belongs to this cell because all the RNA from these cells have this same barcode. Okay, this is the way to tag each RNA. So that's why it's really important to have encapsulated only one cell, because everything that is encapsulated in the droplets will be marked with the same barcode. Okay, so that's why it's really important to have dissociated all the cells. For single nuclei Ataxic, so the idea is to get the information of the accessibility of the chromatin. For example, to predict the position of transcription factors. So you can do this at the level of the cell. So maybe you will identify some subclusters of cells that don't have the same accessibility, and that will help you to interpret maybe the transcriptome you have for these cells. So I will pass on this. Yes, so it's the same principle. All the accessible fragments from the nuclei are tagged with the same identifier. So again, that's why you need to be really careful in the here. You need to encapsulate it just one cell, because everything that is in the droplets will be marked with one barcode. So in this example, you want to identify differentially accessible regions between cell groups. So here you have the list of cell groups, and maybe you can, try to explain why some regions are accessible in some cell types, but are not accessible in other cell types. So that's the way you are using single nuclei Ataxic, maybe to identify this cell type based on gene regulation, like promoter, enhancer, or repressor. So it's like an Ataxic experiment you're using, to detect some peaks, some regions that are accessible, and try to link this to a cell type. To perform single nuclei Ataxic, there is a package, R package, that is the gold standard called snATAC. It was developed by the same team as for the single RNA-seq, so it's from the same developer as SERAT. So I put here the link for the tutorial, because today I would just focus on single cell RNA-seq, but as with SERAT R package, everything is really well described on how to do this. So there is not a lot of paper using this type of data for now, but it's starting to be used by people. So you will see maybe some interesting paper in your field. So as I told you, you can also use the same cells to capture the two information, so the transcriptome and the accessibility. So that's something that the Chromium can do now. So at the end, you will be able to link together the expression and the accessibility. So here, just two slides about immune cell profiling. So that's something, it's another protocol available. So in this case, you can capture B and T lymphocytes to see the repertoire at the surface of the VDG. So you can associate this with gene expression. So again, you have two layers. You have the VDG repertoire and the gene expression, and you can try to link these two information to explain your biological context. So this is based on specific antibody for cell surface. So there is companies for that, and you need to choose which antibody will be used to target which cell protein surface. Yes. So in this example, you can see that you can see at both times the expression and the protein surface for the same experiments. So you can identify, for example, the different subtypes of T cells and see the expression, but also validate maybe the protein surface here to be sure of the classification. So that's something really helpful, to see surface protein and expression. So the last option that people are using now, I have done maybe 2 analyses with this option, is the multiplexing of samples. I told you that the cost is high for this type of experiment. So now the cell hashing is an option to be able to put together samples that have been previously marked by ubiquitous antibody. So the biologist is choosing which antibody is used on which samples, and it will tell the bioinformatician what was used for each sample. So you will have to add this information to the pipeline to be able to distinguish each cells, each condition. So you will know, okay, this cell comes from this experiment because the marker at the surface that was used was this one. So that's something you need to know as a bioinformatician if they are doing a multiplexing. So this is really useful. You are going to diminish the cost of this experiment. This is really interesting. So again, I'm not working for 10x, but again, 10x is proposing a cell ranger solution. That's something really cool. It's a free software that comes on their website that is used to analyze all the data coming out as Chromium. So it's you have all the version, whatever protocols you are using, single cell RNA, ATAC, BDG, cell surfax, multiplexing, etc., etc. They have developed all the version of Cell Ranger for all these protocols. So it's free, but you need a computing resource to use this software. You cannot run this on your computer. And it's already available on Compute Canada. So that's something you just have to load this module on Compute Canada and you will be able to do it. It's one command line. If you know how to do it, it could be simple if you have the reference genome. Also to mention that it's, for now, something done by Genome Quebec. So if the sequencing is done there for all single-cell RNA-seq, if you are doing it at Genome Quebec, they will launch the analysis with Cell Ranger automatically. But if you are doing something more complex, like Ataxic or using multiplexing cell hashing, the Cell Ranger will not be done right, so you cannot use this information. But if it's the basic single-cell RNA-seq, Genome Quebec will perform it, and you can download the results at the end. So this software, Cell Ranger, has a lot of options. You can convert BCL file to FASTQ, but if you're using services at Genome Quebec, usually you are getting FASTQ files. It can perform the quantification of transcript for single-cell RNA-seq. It can perform the quantification of accessibility for single nucleotide. It can also combine single-cell experiment. I put RNA-seq, but it's combine whatever experiments you want from the same protocols. And also there is an option to reanalyze if you want to use personalized parameter after the first analysis. So it's a free software. It has a lot of options. But it's not perfect. I will go into detail after. So this is an overview of the pipeline. So you need someone to prepare the samples. I advise to use a molecular biology facility to process the samples, or to be trained by 10x if you want to use this, the Chromium, then the sequencing that can be done wherever you want. And then you can use the services or do the analysis by yourself. But just to mention that it's a field that is evolving very quickly. So there is many tools coming out, like every month you need to check what is new and what is the trend. So at some point it was pseudotime, and now it's velocity. So it's evolving with the time. So there is a lot of packages available. I will pass on that. Okay. So Cell Ranger, it's one command line, and for single-cell RNA-seq, it will do all the steps in one command line. So it will do the mapping on the reference genome using STAR. So they did not develop anything new. They just use what is available out there for RNA-seq, the STAR software. So mapping with STAR, they are performing quality control checkpoints. They are doing the clustering, the dimension reduction, the projection, UMAP, t-SNE, and also performing differential analysis to find markers. So everything is done in one command line. So that's why it's really easy to use it and generate a lot of files. So this is all the output from CellRanger, and what is really interesting as a bioinformatician is that the output from CellRanger can be used as an input in other R package like Seurat, snATAC, and also in Python package like scMP, for example. So this is something interesting. You can use CellRanger to generate all the matrices you need at the beginning, and after that go in specific packages to perform an advanced analysis. But I just want to mention that CellRanger, the 10x are also proposing a software called the Loop browser that is really useful, for the biologist to look at the data. So they are producing a .loop file, and you use it as an input in the Loop browser. So in the Loop browser, you will be able to see the UMAP, the t-SNE, to select your gene of interest, to redefine clusters, to compare clusters. So you can do a lot as a biologist with this, but sometimes you get lost in all of this. But it's something that you can use to be sure that the data you generated is, something useful that you can use for deeper analysis. So what we want as a bioinformatician from CellRanger are these three files. So we are interested in matrices. So we need the unfiltered or the filtered matrices, depending on what you want to do or where you want to start. So we need the barcode file. So this is all the tag that were sequenced in your experiment. We need a feature file that explain all the features that have been captured. So usually it's only gene expression, so you have the list of all the genes that were captured in the experiment. But if you have done protein surface experiment, you have also the antibody that were captured for the same experiment. And the last file is a matrix of the expression. It will tell you which barcode was found in which cells, is associated with which feature, and what is the count for this feature. So you need these three files to go into maybe R. So, okay, I had a slide. So this is the information I just told about Loop browser. So good tool, but limited functionalities for now, but still in development, and they are putting a lot of effort on this, and it's free, donc it's still interesting. So this is more the pipeline for bioinformatician. If you want to do this with R, for example, so you need the three files for the input. And the first step, as always in NGX experiment, is to check the quality of the sequencing. So we need to remove unwanted cells like low quality cells, empty droplets or doublets, and also the dying cells. So you can perform, I will go into detail about this quality control, but this is really important. We need to work on only good shaped cells. So we are going to check this. Of course, when you want to compare cells at this level, we need a normalization step to be able to compare them. So we will perform just a scale and center the data and the log transformation. So it's a basic normalization at this step. So for now, you have maybe 10,000 cells with maybe 20,000 genes captured. So it's a lot of information. So we need to reduce this information. So in order to reduce the dimension of your data, to be able to extract what is really important in your biological question. The first step is to select features. So a feature is a gene, okay? So we need to find the genes that are highly or lowly expressed in a subset of cells. So we will focus at this point on this gene here. So it's called the feature selection. This doesn't mean that we are not going to see the other genes in our experiment. It's just for to be able to produce a representation of your data, we will focus on these genes only. But after, you can go and see what is the expression of the other genes you didn't select at this step. So we select the features, we are doing a scaling, we are performing a linear dimensional reduction based on the feature we selected just before. We are performing the clustering, and at the end, we are going to have what everybody wants, a UMAP or t-SNE, so it's a representation of the data. But we have to do all the process before to get this type of representation. And after that, you can start the differential expression analysis, and maybe after the differential expression analysis, you will go back to the clustering because maybe something is wrong, you want to merge some clusters or you want to divide some clusters. So you may have a loop at this point to refine your clustering. But these are all the steps you are going to see today about single-cell RNA-seq. So I was talking about quality control. This is a mandatory step. We need to work with reliable data. So of course, here it's really important to talk with the people in charge of the sample preparation and the biologists to find out if any problems occurred during the sample preparation. So you need to talk with them and see, okay, what was, especially with the biologists, if there is any issue at some point in the protocol cell cycle. So maybe the cells are not synchronized. So if it's something that is not interesting in the biological question, you can regress this information from the data to focus on other genes than the genes involved in a cell cycle. You also need to remove doublets, so the droplets that has more than one cells. And also you can check the health of the cells. So for this, you can look at the mitochondrial RNA levels. If it's high, that means that the cell is dying. So for all these metrics, you have to do two things. You need to look at the summary that is generated by CellRanger, first step, and then check out other metrics with R. So for this, we will use the Seurat R package that is really well developed. There is a lot of tutorial available online with ready-to-use datasets. So just copy-paste the command line so you can analyze available datasets. So if you want to be trained, technically trained, you can try these protocols. Also, it's a large user community, so if you have any issue, you just ask a question, or you will find it on the web. Someone already asked the question. Also, something good with Seurat is that there is interoperability between Seurat objects with other package like SCAMP or Loom and other package in Bioconductor. So now Seurat objects are a standard in single-cell experiments. So everybody is using this as an input to develop new package. Also, it's a package that is still under frequent development. So the version is still are still coming up. So they are improving the method behind the Seurat. So for the quality control, as I said, the first step is to look at the general check, the general quality of your experiment with the CellRanger report. So you have on the right here one summary from an experiment. So it's an HTML web page produced by CellRanger. So you have a lot of information, but I will just focus on the most important ones. So the first one is, what is the estimated number of cells? So this should be the target that the biologist wanted. Okay, so if it targeted 10,000 and you just have 1,000 cells, something went wrong in the experiment. So you need to check that the number of cells obtained correspond to the number of targeted cells. So this is the first thing to check here. Then you need to check that you have the mean reads per cell that is recommended by 10x in this case. So it should be higher than 20,000 reads per cell. And of course, if the user wanted a deep sequencing, you need to check that if they ask for 50,000 reads per cells, that you have a number here that is close to this target. Also, for the mapping, so as I told you, they use the STAR software for the mapping of the reference genome. You need to check how the reads align to the reference. So here it's more than 90% of reads align to the reference. So everything is good here. And the last thing to check here is the fraction reads in cells. In the cells, we want this to be higher than 70%. If it's lower, that means that you have ambient RNA, so that the cells were dying, the cells are dead, so the RNA inside the cells were released in the sample. So when you sequence this, that means that you have free RNA. So if you have free RNA, you don't know from which cells it comes from. So that's why we need to this number to be really high to be sure that this reads comes from one cell and was not just a free RNA in the experiment. So this will reflect the mortality, the level of mortality in the experiment. So you need to check this here, the barcode rank plot. This is used to be sure that you have true cells in your experiment. So there is a documentation on the 10x website. But the idea here is to have a knee plot. So that means that you should have something here like a plateau and then a knee. That means that all of this is true cells. So in this one here, we see that we have a knee, so everything seems to be good in this experiment. So that was the first step with the summary from Cell Ranger. So if you have any issue at this point, maybe you don't have to spend more time if the quality is not enough. Okay, if you are just 200 cells and you were expecting 2,000, maybe just don't spend times on bad quality experiments. So sometimes that's why it's really important to check this HTML report to be sure that the data works the work, the bioinformatician. So after that, you can import the data in Seurat, so using the R. So you can do quality controls, more in-depth quality controls. So you can check first the quality of cell isolation. So as I told you, the principle of single cell experiment is to be able to individualize a cell inside a droplet. So to detect these outliers, so if when one cell, more than one cell was encapsulated, this means that maybe you will have twice more RNA in this cell. So this is what you have here. That means that here you have a number of cells that have maybe here three times more end feature RNA than in other cells here. So you see that here you have outliers, so you can use threshold to remove these cells. So you can say, okay, I will keep all the cells with end feature RNA lower than 4,000, And also, I will keep only the cells that have an N count RNA, so it's a number of reads in this case, higher than 20,000. So this will allow you to discard all of this, because this might be multiplex, so you don't want to use this in your analysis. So you can do it with just some plots using the feature scatter function in R, or you can use tools. I never use them, but I know there is available tools to detect such outliers. So the idea here is to detect either empty droplets or low quality cells or doublets. So this is really important to do these graphs at the beginning of the analysis. The next step is to check the health of the cells. For that, we can use markers, and the markers are the genes from the mitochondria. So we can use the level of RNA from the mitochondria to see if the cell is healthy or not. So for that, in this example, generally we will put a threshold. That means that we will keep the cells that have a rate lower than 20% of mitochondrial RNA. So this is in this case because we see that most of the cells have a percentage around maybe 5% here or 10%. So by doing this, we are going to remove all these cells from our data. Okay, so we'll focus only on these cells here. But sometimes 20% is not the right fit. So in this example here, I have the violin plot for another experiment, and you see that the scale is really different. If I put 20%, I will not remove any cells. I have few cells here, so maybe I can just drop the rate, the threshold at 5%, and say, okay, I will keep all the cells with a rate lower than 5%. And in this case, you see that the quality is really good. The level of our mitochondrial RNA is really low in this case. So that there is no rule, but usually we want less than 20%, but sometimes we can go lower than that depending on the quality and the experiment. So now that we have good quality cells, we can start the analysis. So the first step is the normalization. So Seurat is offering two options for the normalization. The first is with the normalized data function. It was the first function they developed. So in this case, they were normalizing the data relatively to the total expression in the cells. And they were just multiplying this by a factor of 10,000 and then apply a log transformation. Okay, this was the first way to normalize the data proposed by Seurat. So then they developed another option called the SC transform. That is a more sensitive method to find biological viabilities, and it will perform in a single command all the data for. We will perform all this function. So it will normalize the data, scale, and find variable features in the same command. So it's another way to perform the normalization. We are in our facility. We are mostly using the SC transform, but sometimes, depending on the data, we see that the normalized data is better. So it's up to you. So we have good data. We have performed the normalization. So now we can start to identify the highly variable genes to be able to reduce the dimension of the datasets. So in this way, we can identify genes that are highly or lowly expressed in a subset of cells. So that way, we will remove all other bias. So we will focus the analysis on these genes because these have been demonstrated to help to find the biological signal in the data. If we keep everything, sometimes it's really difficult to just extract the knowledge from it. So there is different ways to detect these highly variable genes. The default option in SERAT is to keep the 20,000 most variable genes. So based on the standard deviation, they just ranked them and took the 2,000 first ones. So this is one option. You can also keep all the genes. That's another way to capture all the information. Or you can do it by removing bias in your experiments. For example, if you know that the ribosomal level is really high, or that you have still have mitochondrial RNA, or that the cell cycle is doing is highly highly present in your dataset, you can remove this from the list. So it will keep the 2,000 most variable genes, excluding genes associated with this process. So we mostly use the default in SERAT, but sometimes when the dissociation of cells, when we have the projection is not really easy, we go back to this and we try with all the genes to see if we had more information, we are able to more individualize the cells. So now that we have selected this feature, we can prepare for the data reduction. So for that, we will center and reduce the datasets to give an equal weight in the following analysis, so that highly expressed genes do not dominate the results. So we want them to have an equal weight in the datasets. So we can use the scale data of SERAT to do that. So what the scale data function is doing, so it will shift the expression of each gene, so that the average expression in all cells is 0. So we center the expression on 0, and then we shift the expression of each gene so that the variance in all cells is 1. Okay, that's a basic principle. So after the centering, reducing of the data, we can perform the PCA, so it's a principal component analysis. So we are performing this on the previously selected genes. So it was maybe on the 2,000 most variable genes, or maybe on all genes by removing cell cycle, for example. So after a PCA, we need to choose, how to reduce the dataset. So which principal components we will keep for the next step. So we need to decide how many dimensions we will keep. So to reduce the technical noise of single-cell experiments. So we want to focus on on which dimensions explain more the variation observed in between the cells. So why we need to do that? Because this will impact the clustering of the data. Okay, so we need to see which information we keep for the clustering. So how to choose? So there is one thing we can do. It's an elbow plot. So it's another SERAT function that will draw this type of graphs with the PC and the standard deviation associated with each PC. So it's a basic PC here. So you know each PC, what amount of standard deviation it will explain. And using this plot here, we just go to the bottom of the elbow here, and we say, okay, maybe we'll cut just the 10 first dimension in the next step for the clustering, because the 10 first dimension seems to regroup almost all the standard deviation. Okay, but one rule we have in a single cell analysis is that it's always better to have more pieces than not enough. Okay, so if I have a choice to make, I will maybe prefer to do 15 pieces than 8 pieces. Okay, to be sure that I have captured all the important information in my dataset. Okay, so that's the rule. Better to use too many pieces than not enough in this type of experiment analysis. So okay, so decided, okay, I will keep the 10 first in this case. So this will be an argument in the next analysis for the data visualization, for example. So if you are here today, I think you have already seen UMAP and t-SNE. So that's something that is still, a question. What is the best representation? And my answer is there is no best representation. Both representations are good. It depends on the biological information, the biological experiment. So we are using this type of visualization to group together cells whose transcriptome resembles each other. Okay, so that's a way to regroup the cells that look like each other. Okay, so there is two ways: the t-SNE and the UMAP. The main advantage of the UMAP is that it's faster method than the t-SNE, but now with the computing power we have, I don't think it's still an issue. But the main advantage is that it preserves better the structure of the data. Okay, so it reflects more how the cells regroup in the space. But as a service, we are still producing the two representation, and we always ask the biologist which representation has more sense to you. Okay, depending on how you know your condition, your experiments, your samples, because the two are for us as a bioinformatician it's the same. I prefer UMAP, but sometimes the t-SNE will reflect more the biological context. So it really depends on the subject and on the data. Also, as an advice as a bioinformatician, the projection you will make with with a SERAT, if you are doing it later on the same dataset, and if you didn't put a seed in the argument, you will obtain a different representation. Okay, it will look like it, but it can be reversed in the space and etc. So it's always better to put a seed when you are drawing UMAP and t-SNE, because if you want to do it later, it will look differently, and if you are working for the publication, you will have to redraw all the UMAP and all the t-SNE for maybe the markers, the different projection you have put. So this is really important. I have done it so many times. Just put a seed on this to be sure that you obtain the same UMAP and save our object frequently. So just to compare a little bit t-SNE and UMAP. So t-SNE projection is based on one parameter called the perplexity. So it's a parameter to balance the global and the local aspect of the data. So the global aspect is the distance between groups of cells, so it's between the future clusters. And the local distance is the distance between the cells of the same group. Okay, so you need to play a little with this perplexity parameter to be able to get the good representation of something that looks like a good representation of your data. Something really important in t-SNE is that the distance between cell groups should not be interpreted. Okay, that's not because cluster 1 and cluster 4 are distant. That means that cluster 1 is closest to cluster 2. Okay, you cannot interpret the distance between the clusters. It's a projection on a plan of a 3D object. So be very careful with this. For UMAP, there is a lot of parameters, but I will just explain two of them. So on the right here, you have a representation of one data set using pairwise parameter, so the mean distance and the n neighbors. So you see that the parameters have an impact on the aspect of the UMAP. So the first parameter is the n neighbors. So it's the approximate number of neighbors that are close to the cells. It will make possible here to control the balance between global and local data structure. So if we see here, if you have a low value of n neighbors, so at the top left, it will go in favor of local structure, so the cells are really compact here. And if you have a high value of n neighbors, you see that the cells from the same origin, the same cluster, are spreading in the graph. So you see how it impacts the projection. For the mean distance, it's a minimum distance between 2 points, so between two cells. If the value is low, we see that the cells are closer together, but if the value is high, it will keep the cell away. Okay, so it's a balance between these two factors that will impact your UMAP at the end. So just to show you how you can have different projection. Here it's the same data set with the same quality control, same selection of variables, but we have performed two projections, t-SNE and UMAP. So you see that with the UMAP, the cells are more close to each other, so we are able here to distinguish more the different cluster. And for the t-SNE, we see that the cells are spreading more inside the same group of cells. So it's a different way to look at the data. So maybe, depending on the context, the t-SNE will be better or the UMAP will be better. Okay, so now we have the projection, the t-SNE and the UMAP. So we can perform a next quality control. So we cannot do this before we have computed the projection. So now that we have it, we can check for the cell cycle, for example. So why are we checking on this? Because the cell cycle, you can see that in your experiment, the cells are not synchronized. So you can have cells in the G1 phase or in G2M or S. So that's something that can be interesting depending on your biological process of interest, or that's something that can annoy you because you want to synchronize your cells to be able to compare them whatever phase of the cycle they were. So we can check this. So for example, here on this projection, we see that there is a strong impact of the cell cycle. We see in green here all the cells in G2N, in blue S, and in red G1. So the projection of the cell is really based on this cell cycle. So I remember that for this project, this was the goal. We need this information here. We need this projection to be able to identify the cell population, because the cells in G1 belong to one subpopulation. The cells in this G2M are in another subset. So in this case, I will keep it. But if it's not something interesting, if you want to remove this, we can use a regression to remove this information here. Here in another example, we have a small impact of the cell cycle. So we see that all the color, the different cells, the G2M or G1 or S are everywhere in the UMAP. So there is no bias here for the cell cycle. So my advice, if you see a small effect, is to not use a regression to modify your data. Okay, it's always better to alter the data as little as possible. Okay, because you don't know how it will impact other genes, maybe. So if you need to regress this here, it's really easy. In the snATAC package, they already know the list of genes associated with the cell cycle. So they will just use this information to subtract this from all the data to be able to compare the cells regarding the cell cycle. So that was the last quality check. So now we can start the identification of clusters. So at this point, we just have a UMAP, okay? I don't know how many clusters, and I don't know what are these clusters. So the next step is the identification of clusters. The identification is based on community detection approaches, so it's based on graph theory. So it's pretty straightforward, I will say. But there is only one parameter you need to choose. It's called the resolution, and it will impact the number of clusters obtained. So that's something really important here. So you can use the default resolution parameter, and then try maybe two other resolution, maybe increase or decrease this parameter, and see how it impacts the graph. So maybe after the first clustering and after an in-depth analysis of the datasets, you will return to the cluster analysis and maybe choose to bring together clusters or try other parameters to separate groups of cells, because you know that based on the markers, these cells should be separated, for example. So sometimes it takes a long time and a lot of iteration before finding the right division, of cells on this projection. So for this, there is two functions in snATAC called find neighbors and find clusters to be able to associate each group of cells to a cluster. Okay, so now I have a UMAP or t-SNE, I have the clusters, but I still don't know what are these cells. So the next step is the cluster annotation. So my cluster are named from 0 to maybe 14, and I want to know what is maybe the cluster 1. So to do so, there is 3 options available for now. The first is to use known markers or signatures. So this can come from publications, from your knowledge, from the biologist, if he knows that this gene should be expressed, combined with another genes, etc., etc. So at this point, this is the first step you can do, is to discuss with the biologist, or if you know that you are working with lymphocyte, go find the publication and see, okay, this one, I need to use this gene, etc., and draw some feature plots. This can be really time consuming, so of course there is other option. The other option is to use an approach to compare and to detect new markers. So you can try to identify markers for each clusters on a pairwise way. For example, you can compare cluster 1 and cluster 0, or cluster 1 against all the other cells to see what is specific to this cluster here. What are genes are upregulated only in cluster 1 or downregulated only in cluster 1, for example. So, or you can say, okay, I want to analyze clusters 6, 3, 1, and 8 here. So I will compare 8 against all these 3, 1 against the other 3, and etc. Because maybe you know that this is a subtypes, interesting subtypes. You can go, layer by layer. So you can search for markers, and then if you know some of them, say, okay, this one is CD4 T cells because these genes are expressed. But usually, as a bioinformatician, you don't know this. So of course, they have developed our package for automated annotation, and I will talk a little bit about this because I have tried it, and in some cases, it's really a good. They have a really good performance. So if you use a non-markers of signature, usually the biologist is giving you a list of gene of interest, and you just draw the feature plots. So here I have my projection, the t-SNE, and 3 markers. So I will say that these 3 markers are expressed here in this cluster, so I can associate these cells to this subtype. So this is really straightforward. Or you can try to combine these 3 genes here, 3 markers, to compute a score. So it's based on the average expression score for a set of genes with the function called add module score from the R package, RSeRat package. So for example, you can have a signature for effectors, a signature for naive. So the naive are here in red, for example. You have here markers for Tregs, so you see that it's more expressed here at the bottom, and etc. and etc. So you can use your own signature using a list of genes and compute here a score. That's also something you can do. So sometimes you don't have anything, you don't have any markers, so you can look for these markers. As I told you, I can compare clusters against all other clusters, or pairwise in a pairwise way. So doing this, you can use the find markers function that is based on, there are several methods for that. I think I'm saying just after. And it will just give you a list of genes, so a list of markers, adjusted p-value, and something really important here in single cell is the percentage of expression. So you will have PCT1 and PCT2. PCT1 will give you the percentage of expression of these genes in the first cluster, for example, in C4 here. So C4, it means that almost all the cells are expressing these genes, compared to here C5, where maybe half of the cells are expressing this gene in the cluster. Okay, so this is really important, because if you find the marker that is lowly expressed in your cluster of interest, how you can trust that? So you need this to be at least, I think, at 50% of cells belonging to the clusters. Okay, that's why the clustering is really important, because if there is a subset inside your cluster, it might be hard to find markers. Okay, so you need to filter this list here on adjusted p-value, of course, and also on the percentage of expression inside the cluster of interest. So the first one. So it's like in DSEC, after an RNA-seq experiment, you have the adjusted p-value. You can filter on log4 change, on mean expression here in the percentage. So it's basically the same. You have a list of genes, and you need to filter it to decide which are the markers. So there is several methods available to perform the differential expression analysis, so to find markers. By default, is the Wilcox test. I have tried to use DSEC in small data sets, so it's working. But I have found that with a huge data set, so maybe with 40,000 cells, I wasn't able to use it properly. Okay, it was really slow, and I wasn't sure it will add another layer of information. So a lot of options. So maybe it can be interesting to look at, maybe 2 options, MAST and Wilcox, and see how it works, how many markers are in common between different options. So it's the same as in our RNA-seq analysis. So we recommend to use at least two approach, maybe DSEC2 and HR, and see what is common to continue the work only on the common differentially expressed genes. So the last option, as I told you, is to use automated annotation. So I have used this on a project for an atlas of the lung. So the idea was to detect all the cell types found in lung So for me, it was a lot of information to grab. So it was really good to have this option here. So the first step is to choose a reference. So you need to choose properly your reference, because I don't know, if you're working with lymphocytes, if you choose a dataset that is not including lymphocytes, it will not work. Okay, so look at the documentation. So there is several options. I just mentioned two here: the CellDex and the single-cell RNA-seq that are a repertoire of markers that are obtained from RNA-seq, microarray, and single-cell experiment in human and mouse. So if people are doing more and more experiment, it will help here to build these data sets to be able to use it in automatic annotation. So choose properly the reference. So some of them has a finer annotation. For example, here in the Nover Ashton hematopoietic data here, you have, the GO number, the main label, so it's a general annotation, and sometimes you have a more precise here annotation. So you can have naive or mature B cells. So you see here, you can search for the population you're looking for, and if it's in the dataset, it's a good idea to use this to predict this in your datasets. So you can also draw this type of heat map based on the annotation. So you see for each for each cluster, you will see the annotation, the highest score. So you can decide which annotation is the good for each cluster, for example. So this is working for I repeat, this is working, this type of approach is working if the data you're working on have been already published or already studied, okay? If you work on something really new, the data available here to use as a template, to validate your signature will not work. So it works with a general question, but if it's too specialized tissue or cell population, it will not work for now, but maybe later someone will add a signature that you can use. Also to mention that you can add your own signature. So in a single R, you can add your result from an RNA-seq you haven't published yet. So you will be able to use it with the single R package. So now that I have annotated my cells, I know the markers, so I know which gene are differentially expressed, but it doesn't tell me a lot. So I want to add another information that can be the functional characterization. So we use the same tools as in RNA-seq for now. So we like gprofiler. So we are using it in R. There is a package for that. So with a gprofiler, you can perform enrichment analysis with gene ontology, pathway, reactome, transcription factors. I think there is something with the motif, with the disease. There is a lot of resources. So it's really interesting. So we just pass the list of markers or differentially expressed gene or differentially accessible regions. So it also works for single nuclei attack. And you can have more information about the function, about the process, and help you to summarize the results of your experiment. I think I will finish on the data visualization. So that's something people are asking a lot about how to visualize the data. One advice I can give you is that I'm asking people to send me example of what they want, because they have seen in a paper and they like this type of representation. So it's more easy, instead of presenting a lot of representation they don't want, maybe just ask, okay, what do we want exactly? You know, we can put, I can draw a violin plot or a bridge plot or whatever you want. Maybe sometimes it will cut you sometimes. So the basic representation is a feature plot. So a feature is maybe a gene, in this case here. So you can show the expression of a gene on the projection, or you can show co-expression of genes. So in this case, we have two genes and we want to combine this on a score here. So that's another way to represent the data. Also, you can summarize the information using violin plot for the markers. So sometimes it's interesting to see how the expression is distributed in the different clusters. So we can it can help to say, okay, I will maybe regroup this cluster, or maybe I found that there is a subpopulation in this cluster that is expressing a lot this marker. So maybe I will go deeper into this. So that's another way to represent the data. And the last one that we use a lot is the heat map. So we use it even before sending the results to the researchers. We use it to check a little bit about the clustering. So I put an example here. So you have the level of expression of markers. I think it's the top 10 markers for each cluster here. So here, if you look at cluster 0 and cluster 1, you see that the profile is the same. Okay, depending on the list of genes, we have pretty much the same profile. So for us, it's an indication that maybe we need to aggregate this cluster to form a new one. Okay, maybe it's not two separated cluster, maybe it's just one. So you can either go back to the clustering and play with the parameter to decrease the number of clusters, or you just can decide that I will reassign the identity of this cluster to 0. So every cells here will be cluster 0. Okay. So that's my interpretation here. I should merge these two. And if I look at other clusters, you know, I see that there is specific markers to each one here. We have like a scale. So I will keep this separated. So that's something also interesting. So you can draw a heat map based on markers, top 10, top 20. You can draw a heat map based on a list from the researcher. Whatever way you have to look at this, you can look at heat map for the mitochondrial RNA, from the cell cycle genes. So you just choose the list of genes you want to represent here. Another way to look at data, and I found it interesting, it's the ridge plot. This type of representation allows you to find subtle changes. For example, here, if I look at the CCL5 gene in the platelet, we see that we have different level of expression. So some cells are expressing this gene at level 5, at 3, or 2. Okay, so it's not that it's expressed at the same level in all the cells, comparing to maybe this, maybe another example. This gene here, Liz, in this population is expressed, you know, normally in all these genes. Here we see that you have different level. Okay, so like the violin plot, I prefer this one, but it's another way to look at, you know, different population of cells expressing it in the cluster. So here you see again that most of the cells are not expressing the genes in the naive CD4, but other cells in the cluster are expressing it at level of 2. So that's something also interesting. So again, you choose the gene, you choose the clusters you want to highlight. So that's why it's really depending on the biologist. So what do you want to include in the paper, for example, or in the presentation conference. So that's why I say there is a lot of discussion in between the bioinformatician and the biologist. The next presentation is a dot plot, really easy. But I like it because in this way you can also look at the percentage of expression. As I said, it's important to have a marker that is highly expressed in all the cells, that is expressed in all the cells, not highly, but expressed in most of the cells of the cluster. So you can look at the percentage of cells that express these genes and also the average level of expression at both times. So maybe this one is interesting, Liz, because it's expressed in most of the cells from these clusters, but maybe this one is not interesting because the circle is small. That means that it's less, less than 25% of cell expressing this marker. So this can help you to filter your list of markers. So with this presentation, I show you a lot of option, a lot of function from our SORAT. So here, just an overview of all these functions, the main function, I would say. So of course, there is a function at the beginning to import 10x results. So it's called read 10x. So it can be from the 3 files I told you, all the matrices, or it can be from the H5 files. So it will allow you to import the results and then to create the SORAT object. So I will just detail after this specific object. For the normalization, 2 functions available: normalized data and scale data. All the graphics you can generate. All the option for the dimension reduction, either run PCA followed by t-SNE or run PCA followed by UMAP, or both of them. And finally, all the statistical function to find either variable genes, neighbors, to be able to find the clusters, or to find the markers. Okay, so these are the main functions. There is a lot of functions, but these are the one you need to know if you want to start to use SORAT to do the analysis by yourself. So yes, so 3 years ago, I think we started to analyze the single cell experiment, and we discovered the our SORAT object, and it's a complex object. But once you have understand how it works, it really makes sense. Okay, so just here is an overview of what is inside this object. So it's when you use R to analyze your data, you will fill this object during the analysis. So at the beginning, it's an empty object. When you use the create SORAT object, it's empty, but all the structure is here, but the information is not there. So it's an empty. So on the initial import, you will have 4 categories in your SORAT object. The first one is for assays, so it will be filled with the row counts from the matrices you provided, and after the normalization, it will add the normalized data. Then you have the metadata structure for the experimental condition. So you can have maybe 3 or 4 experimental condition, different metadata for the experimental condition. So maybe you have one annotation for maybe the drug used, one annotation for the gender of the samples, maybe one annotation for the time of the sample. So you can have a lot of experimental condition at this point. So also all the metadata regarding the QC metrics will be added at this in this metadata structure, and finally the clusters information. So you can have the clusters, the initial clustering, maybe after we have the second clustering, the final clustering. So you can name it as you want, and it will just keep it will keep the former information in your SORAT object. Okay, so you can have multiple annotation, multiple cluster annotation in your SNATAC object. So the metadata structure is really important because you can keep all the information you want in this one. The next one is embeddings, so it will store all the projection information, the PCA, UMAP, and TSNE, and again, you can have the initial UMAP, the UMAP with another perplexity parameter, and etc., etc. So you don't have to erase the former information. You can keep all your tests in the same object. And the last one is the variable features. So it's the list of the most variable gene used for the projection. Yes, I think I have just a few slides about. Okay, all I've said before that is about analyzing one samples, but how to analyze multiple samples. So if I am doing maybe two samples, one is wild type and the other one is knockout, but I want them on the same projection. There is two options. So the two options are integration of data or aggregation of data. So the aggregation allows to combine different objects together with the normalization on depth. Okay, so this is an option in cellRanger. There is a cellRanger aggregate function, and it will subsample your data so you can compare them together in the same space. Sometimes the aggregation is not the best option, so you can try the integration of data because it will correct for biases like technical bias and also biological bias. But when I say that it will correct for biological bias, sometimes it will remove too much biological bias and you will lose the information you want to extract from the difference between a wild type and a knockout. So it's not always the best solution. So sometimes we go for the last solution, I didn't put it on here, is just to merge the data in SNATAC. Okay, so it's not an aggregation with cellRanger, it's just to merge at some point the data together. There is a function in R SNATAC to merge two datasets together. So this way, there is no subsampling, there is no removing of batch effect. So how to choose? I will say it depends on the projection you obtain. So you can try aggregation. For example, here at the top, we aggregated knockout and wild type data. And we have here data without bias. So we see that the two projections work well. Here at the opposite, we have aggregation of data, and we see that the projection is on one side the mouse and on the other side the human cells. So the differentiation here is based on the sample, so we cannot compare them together. So in this case, we need to use either integration method or try aggregate. We need to use integration for this type of data. So this is always tricky. So usually we start with a merge in our experience. We use a merge in SNATAC, and then we check if the segregation of sample is done well, or if we can compare sorry, the cell together. Okay, so there is advantage for integration, of course. So it will usually it will be better to use integration, so you can compare condition, for example, condition 1 and condition 2 here. Here you see that with the same data set, if it's aggregated, we cannot compare them because it's not at the same on the same layer, on the same projection. So there is several methods for the aggregation. So we use the last one from SNATAC, but other tools are available, and they are still working on this. So we are waiting for a new way to perform this, because we have seen that the integration has also regressed out the biological meaning in the datasets. I think I will pass on it because it's almost time to stop. Just to finish, so after you have maybe integrated your datasets, you can try proportion comparison. So maybe you have two conditions and you want to be able to say, okay, in this condition, I have more cells in this, I have more cells from this population than in the other one. So you can use maybe the DitoSec R package. So it would just facilitate the visualization of the proportion of each population. So it's not a statistical test, it's just visualization of the data. So maybe here we see that we have more blue population here in female than in male, but is it statistically meaningful? I don't know. So that's why there is another option called SC proportion test that can allow you to compare the proportion and have statistics about it. So you can have a threshold from the FDR lower than 0.05. So that means that the proportion of the goblets are different here for these clusters between your experiment. So that's a way, to have statistics associated with what you were seeing here at first. So I will finish with the advanced analysis. So people now, they all want pseudotime analysis and velocity analysis. So what is the pseudotime? So the idea here is to reconstruct a trajectory to try to extract, I would say, a process that is continuous during time. So for example, the cell differentiation during development. So if you're working in this field, it's really applied to you, pseudotime and velocity, because the cells are changing over time. So there is several software to do so. We have this in R in Python. So the initial package was Monocle, but now people are also using Slingshot or Velocito to produce such analysis. So in this example, Monocle, I'll just show you an example. We have here a UMAP with, you see, a lot of clusters, and we want to understand how the cells evolve through the process. So we have C. elegans neurons during embryo's development. So we can use Monocle on this. So we tell Monocle that the root is here. So the root is the initial state of differentiation. And then Monocle will found by itself the different option, the different.
Live streaming is on.
Okay. So Monocle will identify the pseudotime associated with each cell. So here you see the gradient, blue for initial pseudotime, and here a more advanced state. So here, if we start from the root, you see that the cell evolves. Here you see the pseudotime is increasing, and you have here branches. So that means that there is 6 fates possible at this stage. So you have the bifurcation, and your cell can end up here or end up here. So that's why we use pseudotime to see, how your cell evolves and what are the fate available in this process. So based on this information, based on the pseudotime, you can now compare these cells here. Maybe say, okay, I will take all the cells with the pseudotime between 12 and 14 here, and I will compare them with the previous one, okay? And I will perform a differential analysis and see, okay, what has changed between the two, okay? During this time, what has changed? So you can now perform a differential expression analysis based on the pseudotime, not only on the clusters, but based on the time. So this way, maybe you can see this type of trend. So for this gene here, we see that the expression is increasing at time 8, I would say, and then going here to a plateau and decreasing. Here, on the other hand, you have two other genes with pretty much the same curve, and the expression of this gene is starting here around time 11, okay, in the cells belonging to cluster in green, and then decreasing. Okay, so this is another way to look at the data, to add this information and be able to, identify the changes, the small changes based on the pseudotime. So that's one way to do it, the pseudotime. And the new way to do it is to look at the velocity. So it's not the pseudotime, it's to use another information. Okay, so for single-cell RNA-seq experiment, most of the RNA that you capture are mature, but we have some immature RNA in these experiments. And if you have immature RNA, that means that this RNA was just transcribed. So you can quantify the activity of each gene, okay? So the idea here is to have a matrix associated for introns. So if you have introns, that means that your RNA is not spliced, so the transcription is active. So a matrix for unspliced reads, that on introns, and the matrix for spliced reads, so they are in introns. So that is just a way to use the information inside your data. So we use the BAM files, so the BAM are generated by STAR. We use them with the Loom R package or whatever way to compute these unspliced and spliced matrices. And then Velocito, a package available in R and in Python, will be used to compute this velocity, so this level of transcription. So I would just put one figure here. You can produce a lot of representation with Velocito package. So here, for example, we have two clusters, cluster 10 and cluster 5. And we see that the arrows here that represent the direction, where the cells are going to, okay? So that means that the cells in cluster 10 are tending to become cells in cluster 5, okay? So as a bioinformatician, I cannot say that, but biologists analyze the data. Cluster 10 is epithelial cells, cluster 5 is podocyte population, and they know that cells in 10 will become cells in 5. So that's a way, to interpret these arrows to say, okay, how the transcriptome will shift. So how the cells will evolve to another stage. So also something really interesting. And this cannot be done with SaveRanger or whatever user-friendly tools for now. You need to know how to code in Python. Even in R, it's not that easy. So with Python, it's more easier because we need computing power for such analysis. So that's something we are doing now for like everybody on a Velocito analysis. But sometimes it's difficult to find biological meaning in this. But still really interesting. So I will finish on this with the take-home message. So if you want to perform single-cell experiment, you need a good planification. Talk with a lot of people from the molecular biologist, bioinformatician, prepare yourself for such experiments. Also, depending on who you are, biologist or bioinformatician, just know that you will have to work in close collaboration together. Because you need each other to extract information from this type of data. So this is really important to have someone that you can just, send an email and be sure it will answer you in few days and not in weeks. Really important. And my last advice would be to go step by step during this type of analysis. Do not jump directly into pseudotime or velocity. Just look at your data, know your data sets, know your cluster, what you have, what are the issues before doing such advanced analysis, because you will don't know how to interpret the result if you don't know well your data sets. So thank you for your attention. I hope you enjoy this, and if you look at this later, if you have any question, you can also ask me, and I will be happy to help you with this type of analysis.
Thank you, Virginie, for your presentation. It's very, very instructive, and thank you to educate us on single cell.
You're welcome.
I don't know if you have question.
I don't see any question on the chat.
Me neither. Neither in YouTube channel.
Okay.
Okay. But I have a question. If you go to the slide 54 of data visualization, I saw.
I closed it, I think. Okay, go ahead.
Can you explain about the legend?
It was the feature plot, or
yes, I think so.
Okay, it's open. You mean on the?
And can you share your slide, because we didn't see?
I'm going to try.
Thank you.
Okay. Yes. So the legend on which one?
About the color threshold. The
on the right.
Yeah, the heat map.
So here it's based on the expression of the two genes here. So if you have the same level of expression in the two, the color code will just use, if you have 5 in CD, you will have this color if you have 5 also here. So it's just based on the expression. So if it's more red, that means it's more expressed in this one. If it's more green, that means it's more expressed in this one. It's like in microarray expression. And if you have something like in gray or yellow, I would say it's the same level of expression for both genes.
Okay, perfect. So if it's gray, it means that it's not expressed for the two.
Yeah, that it's more expressed in this one, or less in this one.
Okay, perfect. About the page 46, you said that we need to play the parameters to know what's the best resolution.
Mm-hmm.
But is there a threshold, or is there a mathematical equation that we can say, we need there, or it's based on data visualization?
Yeah, so it's based on the visualization. So usually you start with the default resolution, value, and if you increase the resolution and you see that you don't have more clusters, you can say, okay, how to say that? The community detection is working well. It's not because I'm increasing the resolution that I will have more. Okay, if the data are strongly connected, it would not divide it easily. So that's one way you can decide to increase or decrease the resolution parameter, or you can go to the heat map directly, find the markers, draw the heat map, and say, okay, this way I will say I need to merge cluster 0 and 1, and I would leave this one together.
Mm-hmm.
That's one way to do it. And also for the small clusters here, sometimes we remove them at the end on the UMAP, because sometimes it's just junk cells. We don't know what they are, so we need to remove them if we don't know what they are.
Okay, I see. So it's manuable. You can just play it on the code. Yeah. And you was just showing the package on R. I'm wondering if there is similar package in Python.
SORAT is not available in Python, but there is a way to analyze data with a Python tool. So if you generate the matrix at the beginning, you can go into Python to produce all these steps. But it's a different algorithm than SORAT, but still you can, and it's faster in Python.
Okay, perfect. And on the page 50, you was talking about the percentage of expression. Here you just showing for 2 clusters, but I'm wondering if we have other clusters and we need to see if the cells, the gene is expression on different clusters.
Yeah, if you are performing analysis against all the other cells, the percentage here will be difficult to use, because I guess that in all the cells it will be difficult to reach 50%, for example, of expression. But still, you need to put some threshold on these percentages here, because you cannot use a marker that is just 10% expressed. It's not meaningful. So you can decide here some threshold and maybe draw the feature plots and see if it's relevant or not. It's the visualization will help you also to decide which markers are useful.
Okay, perfect. I understand. That was all for me for the question.
Thank you. You are the one that has question.
Yeah, we are on the time, 6:30.
Perfect.
So if we don't have question, would like to speak. Yes, go ahead, Jean.
Oh, it's about BDG. Yeah, I did not talk a lot about this one. Yeah, the recommendations are different. The one I gave are for single cell RNA-seq. They are also different for single nuclei, Ataxic. And yes, for VDG, you need to follow the recommendation from 10x. So you have a question, Jean, or just a comment on this?
I was trying to unmute you, but I don't know if it's if you can speak.
Doesn't have a mic. So.
Oh.
But if it's Jean for IRCM, you can just ask me tomorrow the question.
Yeah.
Okay.
Sorry. If you have other question.
Okay, I think it's all.
Okay, that's all. Thank you so much. Thank you, Virginie, for your presentation. And see you next. What were the slides? 69, 71. Oh, I have one question from Benoit Pissé. He said, what were the slides of 69 to 71?
Oh, it was about the steps for the integration. So I just described, I just passed through this. I just described the different steps for how the service is integrating data. So the idea is to identify anchors between the samples. So that way you can find cells that are likely to each other in different samples. So that will use this as an anchor to superimpose these two projections, and it will help for the data integration. So it's slide from the integration documentation in Seurat. So that's not something specific I draw.
Okay, so Jean, she asked, could you download your PPT so we can access in...
I don't know. Usually people are sharing the presentation, or...
it depends on you, but we can share it on MonBUG.
Maybe only on MonBUG, yes. But maybe on...
I can share it on MonBUG.
Yeah.
There is one question from Follow Jits here. You can speak up.
Huh? It's me?
Yes.
Hello?
Okay. Yes, it's just a question about the number of cells, because like on each step in the workflow, you have less cells because they are filtered, because of quality control or because of several things. So integration also eliminates some cells.
Sorry, I just don't get the question. Can you repeat, please?
So yes, I know that at the beginning we have almost like 10,000 2,000 cells, for example. Then on each step, after filtering or the multiplexing, a number of cells is reduced. And I wanted to know, is there some way that integration also reduce number of cells?
No, you will keep the same number of cells, but you can lose some information about the level of expression.
Okay.
Yeah, the cells are still there, but the level of expression is just normalized, so you can compare them. Normalized regarding the depth of sequencing, for example.
And that can influence the cell type.
Yes, that's why I said that integration is not always the solution. Because when you are downsampling or integrating data, sometimes you remove the biological information. You know, if it's too difficult for the algorithm to integrate the data. But usually it works well, but sometimes we have seen that it will regress out what we want in the data.
Okay, so that happens just sometimes, not always.
Yeah.
Okay, yes. Okay, thank you.
You're welcome.
We have one last question from Benoit Fissé. He said, leader further page 74 and 75.
Everybody's curious. 74, 75. It was, yeah, the 74 is about the definition of pseudotime. Just I was going quickly on that one. And 75 is how monocle work. So it's just the beginning. I have there is 3 steps for monocle. So the first one is to define cell progression. So it's based on a machine learning approach. So the first step is to select genes of interest. So there is your option to select genes at this step. So it can be unsupervised, so you don't tell anything about the biological process, or it can be semi-supervised, so you can provide a subset of genes linked to the process you want to represent with the pseudotime. So first selection of genes, then it's dimension reduction, and lastly it's to reproject cells according to the pseudotime. So it's just to draw the pseudotime and with monocle. So it's just the monocle vignette.
Thank you, Virginie. Yeah, I think that's all for question. So thank you again for your presentation. And for the next month, we will have another presentation and a meetup in. We go into group in Montreal with all bioinformaticians. So stay tuned and see you next month.
Okay, thank you, Jasmine.
Thank you. Bye.